So hello, everyone. We just heard in the last talk that there are over a million computer structure models, and around 200,000 experimental structures that are now available from the RCSB.org website. So that's a lot of structures, a lot of data. And in order to be able to use this in any meaningful way, the first order of business probably is to find the structure or structures that are related to or relevant to your topic of interest or research project. And then once you've found that structure or those structures, how do you learn or try and understand what this structure is all about? What does it look like? What does it contain, etc.? And then having looked at that, that's what we will cover in looking at the structure summary pages. And then how can using that data help you go forward in your research? We'll try a couple of case studies, and some of these examples will be continued in the subsequent talks. So the first order of the first business that we want to do is to find structures that are related to our topic of interest. And there are a couple of different ways that are available to navigate this large collection of structures and to find things that are relevant and interesting for you. Here we've created an infographic. So I'm going to quickly show you that infographic. This is available on the website, and you will get this link later if you get the PDFs. This describes that there is the most commonly used search option is the top bar search, where on the top of the page, you can type in either the molecule name or any other information that you have about the structure that you want to find. There are also extensive advanced search options that will be discussed in this talk and in the next talk. And then finally, there are these browsing capabilities that we will mention briefly. And then the rest of the infographic actually gives you detailed descriptions of various scenarios. So I encourage you to actually go and look at that and explore it to see what is relevant to your topic of interest. Okay. Now, what I will do is primarily talk about the top bar search and go a little bit into the advanced search options. So just to give you an overview, the top bar search, in addition to typing in a molecule name, a keyword, or a phrase, or specific identifiers, perhaps you are reading a manuscript, and in the manuscript or in the paper, there is a particular identifier listed. You can put in that identifier, or maybe you found these IDs or identifiers in another bioinformatics resource that is relevant to your research project. You can also bring in sequences. So one letter code sequences of proteins, nucleic acids can also be put into this top search bar box now. And this mechanism of this toggle switch, turning it on to include computed structure models, the default being exclude, this we will show you how to use it so that you can, depending on what you're looking for and where you want to go with your research, you can either include or exclude CSMs in your search. The other kind of search that is also available right from this top bar search is the documentation search. So this is not going to return structures or ligands or anything like that, but it will tell you if there is documentation, if there are any news items, if there are PDB 101 related articles describing that topic or that term that you have typed in. And we will look at some examples as we go along. The advanced search is not only limited to textual searches, it can actually also, you can use various attributes that are, or metadata associated with these structures. So these metadata for experimental structures, for example, you can search by resolution, you can search by various integrated other metadata, such as enzyme classification or domain classification, or whether or not it is associated with the membrane. These kinds of properties that have been aggregated from other data resources are also available here for performing your searches. You can use sequence-based searches, 3D structure-based searches, and chemical searches, such as using chemical formula, INCI, strings, etc. And so I'm mentioning this briefly. This will be elaborated further in the next talk. Again, here in this panel, notice that there are these toggle switches available to either include or exclude the CSMs. And the browse options currently are only available for experimental structures, the PDB structures. They are not currently available for CSMs. But these actually organize the entire collection of experimental structures according to various categories, such as whether or not it is involved in a particular biological process. Here we are looking at the enzyme classification organization. So there are that many oxidoreductases, transferases, and you can open up these browser, these tree-like things to find specific structures that belong to a particular class. And then that gets you started on your exploration. So now I'm going to actually try a few examples. So we will do some top bar searches, and you're welcome to follow along. So if you are able to go to the browser to rcsb.org, we can try this together. So let's go for it. So I am going to open a new tab. And here, in the top bar search, I'm going to try a simple search for a protein insulin. So I can type insulin. And as I am typing, notice that there are these suggestions that are being made in a box below. So if I ignore these suggestions and just say insulin and press enter, it gave me some 8,300 and something structures. But these may or may not be all just the insulin molecule. And even the first result shows you that this is insulin-like growth factor. This is not what I wanted. So the better thing to do is when I type in insulin, is look through this suggestion and say that, oh, I want structures whose molecule name insulin is matching, not insulin receptor or insulin receptor related protein or insulin degrading, none of those. I want only insulin. So let's try insulin. And these are now all insulin structures from a variety of different organisms. And I can say, okay, I only want to look at human insulin molecules or whatnot. Now, notice that when I did this search, I just used default options, and the default option was CSM off. So there was no CSM included in this search. I can go back and rerun this. Sorry. Insulin. And before I click on enter or do this, I'm going to turn this on. Now, when I run the search, notice that there are 75 computational structures also in this list. And this is ordered in various, so this is a scoring criteria. You can order it in many other ways. But just so you can look at these, here are some of the insulin CSM structures from various organisms. Okay, so with the name, protein name, I can either select from suggestions or just press enter and run this. So I can also come in and say, I have this particular PDB identifier, for example. And so let's try that. So let's say I know that I want to look at this particular structure. So I can put in the PDB ID. And here is the structure summary page for this. I'm actually going to open a new tab and try this other. So this is a CSM structure, sorry. I'm going to skip out so that I can actually copy and paste. So this is an AlphaFold protein identifier. And I can actually paste that in the top search box. But remember, if I run this search with the CSM off, then I will get no results. So I need to turn that button on and run this search. And I find that structure. And I'm going to open that too. So what I did when I found the 1-TRZ and this other AlphaFold protein, I clicked on that and it opened something that we call the structure summary page. And we will look at that in a few moments. But before we do that, I wanted to also point out that other identifiers, such as identifiers from UniProt, that can also be used in the top search box. I can paste this UniProt identifier, and as I'm typing it, it is asking me, is this what you want? And this one, I can choose to run it with or without CSMs and find results. So there is one CSM structure and 300 and something experimental structures that match that UniProt. So in all these cases, we are finding structures that match that particular query. One more example, let's try this sequence search. So this sequence I got from some other structure somewhere else. I can paste that and run a search. And it found all these different matches. Now, effectively, what has happened is when I pasted the sequence up in this top search box, it actually got automatically transferred into this advanced search query option. And you'll hear more about this in the next talk as to how this space, this advanced search query builder, it actually aggregates information from various places, and it completes your query so that you can, save this query or run it again, etc. So now we have looked at all these different possibilities for running a search from the top search box. What we haven't looked at is this documentation mode. So let's look at that. So here, there's a pull-down menu where I can change the setting to documentation. And now if I type in insulin, I'm not going to get the insulin structures that I was looking at right now, but there are various documents that describe this. There are news announcements where insulin has been mentioned. There are PDB 101 articles. So this is a completely different collection of results that can help you learn more about a topic of your interest if we have data related to that or information related to that. So I am going to now shift gears to the next topic, which is the structure summary page. So we have run a search, we have found some matches or some structures that we want to explore. If we click on that link and open a structure summary page, this is what the structure summary page looks like. I wanted to give you a quick tour of the structure summary page and then move forward. So on the structure summary page, there is, of course, the identifier, some header information giving you some quality metric of the this particular structure. On the left-hand corner, you are looking at a quick snapshot of what this molecule looks like. And the presence of these different colors tell you that there are multiple chains in this particular structure. Information connecting you to the literature. So the primary article that describes this structure is available from here. The macromolecule section gives you a lot of information about the specific proteins, and if nucleic acids are present in the structure, things about that. And if we continue scrolling down further, there is information about small molecules. So this particular structure happens to have these specific ions. And if I click here, I can actually explore the environment around where this ion is located in the structure. And then more details about the experiment and the validation and in entry history. So this is sort of what all structure summary pages contain. These are experimental structures. Now let's look at the structure summary page of a CSM structure. So these structures, if you look at this is not as detailed or informative because there are no ligands, etc. But again, it has the same kind of layout. So in the top left, there is a quick snapshot of what this molecule looks like, and it is colored by that coloring scheme that is explained here. So dark blue or cyan colored regions are highly confident or confidently predicted. The orange and yellow regions are less confidently predicted, and we'll hear more about this in some of the upcoming talks. This gives you a sort of overall estimate of how many amino acids in this particular structure are in the high confidence or middle confidence or low confidence regions, just so you get a sense of what you should do with the structure. Do you want to use this to further explore more ideas? But then below that, there is this macromolecule section that tells you various types of information about the protein that you are whose structure you're looking at, and some
4 minutes.
Yes. Okay, so now that we've looked at the various structure summary pages, we have I'm going to take you through an example where I wanted to show you, like, okay, so you've looked at the structure summary page. So what can you do with this information? So for that, I'm going to take you to one particular structure, 1IEP. 1IEP. Now, this structure is that of an Abl kinase protein bound to a drug called imatinib, or Gleevec, commonly known as. Now, in this particular structure, clearly there is the protein. So here we have information about the kinase. And then there is information here about this ligand, the drug Gleevec. The question that I'm going to ask here is that this particular kinase, does it bind to other drugs or other molecules? How would I figure that out? And so to do that, if I click here, this is a UniProt ID-based search that will pull up all other instances of structures in the PDB that have this particular UniProt ID. So if I click on this, let's see what happens. And here I can already see that, oh, there are these other, so it's the same Abl kinase, but it is bound to other different things. So this was our query. And here are different drugs or other experimental molecules that are bound to that same protein. So this can give us some insights and help us define the next question. What we can also do is ask a different question now. So now I'm looking at the drug, Gleevec. And if I ask the question, does Gleevec bind to only this Abl kinase, or does it bind to other proteins too? And I can run a query on that. And if I just scroll through this, so of course there is Abl kinase, but now we are seeing some other kinases show up. And so this way we can actually, so here Sark and Abl, so there are other things that we can find. So this way we can explore the archive starting from the structure summary page and find interesting pieces of information that may be relevant to our research. Now I'm going to do 2 quick case studies, and that will help you see how having access to CSMs can be valuable in this RCSBPDB.org. So for that, I'm going to go to this PDB entry 1B54. So let's go to 1B54. And the question that I'm asking here is, so this particular structure is that of a yeast hypothetical protein. It has this PLP, a ligand bound to this protein, and it is a model for a human protein. So the question I'm asking is that what is that human protein for which this is a model? So I can do a sequence-based search now. And I'm going to say, I don't know exactly how similar the yeast and the human proteins are. So I'm going to be very generous and look at the, let's say, the 40% sequence alignment. And what I didn't do is I didn't activate the CSM. So I'm going to do that now and rerun that search. And so now notice that the result set has this yeast yeast CSM and many other CSMs as shown by this listing. I am interested in the human protein. So I'm going to open this up and look for the human protein and narrow that down. And so this is the human protein that the experimental structure was a model for. And if I compare this structure with that 1B54, I can learn how similar or different these 2 proteins are. And maybe it'll give me some ideas about what experiments to perform to understand the function or the structural stability of this particular protein. Okay, and this will be revisited in one of the later talks. And the last case study that I wanted to do is take you to one other PDB entry. This is 3CBB. Let's go to that. 3CBB. Now this is the DNA binding domain of the hepatocyte nuclear factor 4 alpha. This is a diabetes-related gene product. And what I'm seeing in this structure is just the DNA binding domain bound to DNA. But I want to see what else the protein contains, where are the other domains, and can I find more information about this? And to do that, what I'm going to do is I'm going to explore this PDB from this structure summary page, this UniProt entry. And if I click on this, I'm going to open up what we call the protein feature view. And here, this top row is an AlphaFold structure of the complete sequence. And then this was our query. This is just the DNA binding domain. And these are examples of the ligand binding domain, the other part of the protein. And so interestingly, here we see one structure that encompasses both the DNA and the ligand binding domains. So we can look at all these, compare them, and make sense of where these different domains are, how are they interacting with each other, how are they interacting with their target molecules, the DNA, etc. And that can help us design or develop new experiments and progress our research. So with that, I think that was our last case. And I just wanted to quickly summarize that we looked at the top bar search, advanced search, browse options. We looked at a couple of query by example scenarios. And we did these 2 case studies. And some of these examples will be used again in some of the following talks. As far as I'm aware, that the PLDDT information is coming from the source where the model was calculated. So we are not computing it, we are getting it from the source.